Hello, my name is Ida, and today I will be discussing the work I performed this past summer in the Bartoli Brain Tumor Lab at Columbia. As entitled in this work, in vivo two-photon imaging reveals infiltrating glioma cells' local effects on neuronal responses to sensory stimulation. Before jumping in, it's helpful to provide some background into the experimental impetus behind our work. Currently, glioblastoma is one of the most aggressive and prevalent forms of primary brain tumors. Glioblastoma has one of the lowest survival rates of any solid tumor, with a median survival of less than 1 year post-treatment. The current standard of care consists of surgical resection to remove the tumor core, which is then followed by chemoradiotherapeutics to try to target the remaining cancerous cells. Notably, almost all gliomas will recur in new diagnoses, and this recurrence generally originates from the infiltrative edge or tumor margin that is left behind. What makes the tumor margin region in glioblastoma particularly interesting is the evident intermingling with a diverse array of cell types found in the brain. In this image, for example, we can see the tumor core as dark orange cells, and these are interacting with neurons, which we see colored yellow, microglia, which we see colored light blue, and other cells in the brain like astrocytes and oligodendrocytes. Interestingly, in our images taken from the glioma margin, we can similarly see glioma cells interacting with endogenous brain cells, including neurons and microglia. So, to reiterate some of the facets that make glioblastoma a particularly complex disease, some of these include 1, the diversity of cell types at the brain tumor margin, 2, the high cellular density intermingling of cell types and proximity that allows there to be pathological effects on nearby neurons, and 3, evidence of brain tumor crosstalk, and this interaction being a crucial regulator of cancer initiation and progression. Specifically, it's been shown that excitatory neuronal activity contributes to glioma growth. These findings necessitate a better understanding of glioma-induced neuronal excitability and the underlying mechanisms. Given our interest in investigating neuronal activity at the glioma margin, we employed two-photon imaging. In this image, you can see how our mice were implanted with cranial windows. In order to do this, the thick part of the skull was replaced with glass, allowing us to measure dynamic calcium changes via GCAM. The bar near the mouse whiskers demonstrates how the mouse also simultaneously underwent whisker stimulation during parts of our timed imaging sessions. The Knoll lab has developed a mouse model that recapitulates features seen in human tumors. So we took Thy1 GCAMP6F mice and cortically injected them with M-cherry labeled glioma cells and then implanted cranial windows. We were able to see an intermingling of tumor cells and excitatory neurons in our imaging field at the infiltrative tumor margin. In this image here, we see that neurons are colored in green and tumor cells with red. Moving forward, neuronal regions of interest and time series extraction from two-photon GCAMP movies was then performed, and all subsequent analysis took place using custom-built MATLAB scripts. Taking 2 sample cells, A and B, from an imaging field, we demonstrate here our event detection paradigm and synchrony score generation. For the top 2 graphs, the y-axis has the delta F over F, or change in fluorescence signal versus time. Peaks or events, indicated with the small gray triangles, were identified by thresholding above the time series baseline. We applied a Gaussian convolution function to the peaks identified in the neuronal time series in order to more accurately and probabilistically determine correlations in synchronized calcium events across neurons. In the bottom graph, we can see the peaks for each neuron after the Gaussian convolution, overlaid on top of each other. The y-axis here being instantaneous events per second versus time. In generating our synchrony scores, we treated a neuron like the center of the universe, whereby we determined an average Pearson correlation value for every neuron based on correlations of that neuron's peaks with the peaks from all other neurons in the same imaging field. This is a representative image for neurons that are highly correlated with an R of 0.82. Taking our recordings from the whisker barrel cortex, we expect to see some stimulus-evoked response. Looking at this graph, we have the average correlation score against the different imaging conditions. The 2 blue violin plots on the left are the control, or no tumor conditions, with no stimulation on the left and stimulation on the right. The 2 violin plots on the right in purple and in purple and orange are the tumor conditions with no stimulation and stimulation on the left and right, respectively. Interestingly, when we looked at the synchrony or average correlations across all animals in each condition, there are minimal differences between control and tumor brain in synchrony correlation values. Seeing this minimal difference between groups drove us to break down the populations of neurons further, expecting to likely see the greatest response in proximal neuronal populations, neurons that are close to tumors, essentially. In this image, you can see that we use neuronal regions of interest, which are outlined in white, and tumor cell regions of interest marked in red and yellow to compute a tumor burden score for each neuron as a function of its distance to tumor cells in the local tumor microenvironment. The graph here, which is taking a second to load, is a histogram with the frequency of all tumor burden scores indicated on the x-axis. Here it is. Sorry, it took a second to load. Notably, there is a bimodal distribution in the tumor burden scores, which allowed us to binarize or bin scores into high and low tumor burden. Looking at this graph, we have the average correlation score against the different imaging conditions. The grouping of 3 violin plots to the left of the dotted line are the non-stimulation condition, and the grouping of 3 violin plots to the right of the dotted line are the stimulation condition. So we have non-stimulation and stimulation over here. Focusing on the stimulation condition, the orange violin plot is the stimulation condition for neurons from control brains that did not have tumor. The red is the stimulation condition for low tumor burden neurons, and the green is the stimulation condition for high tumor burden neurons. Ultimately, the largest effect that we saw after further splitting the data into high and low tumor burden neurons was that in the whisker stimulation condition, the grouping to the right, the dotted line, we saw that neurons with high tumor burden scores in green had higher synchrony scores than the control neurons in orange, and had a higher synchrony score than the low tumor burdens in red in the whisker stimulation condition. And this was all statistically significant as well. Ultimately, we sought to better understand neuronal activity changes as a consequence of the brain tumor microenvironment, specifically by examining the relationship between neuronal activity and tumor burden. When neurons are not stratified by tumor burden, we see that there are minimal differences in correlation values between the no tumor and tumor-bearing conditions. However, we saw that there was a statistically significant difference in neuronal synchrony scores after stratifying the neurons by their spatially defined tumor burden. We saw that neurons with high tumor burden scores had higher synchrony than the control neurons and had higher synchrony scores than the low tumor burden neurons in the whisker stim condition. So our future studies will likely explore the functional consequences of these alterations and synchronicity among paratumoral neurons and their potential link to known findings of altered neuronal and epileptiform activity in brain tumor-bearing patients. Notably, despite recording thousands of neuronal time series, the average synchrony scores remain quite low in both the resting state and whisker stimulation conditions, which we attribute to the broad temporal resolution. Because our results showed the greatest effect in the whisker stim condition when you compare high and low tumor burden neurons, our future directions will focus on the neuron firing during the stimulation itself. Similar to a raster plot, in this graph, we have the time series of 80 neurons stacked top to bottom. Highest tumor burden scores on the top in purple, and decreasing in tumor burden score as moving downward. The red dotted line distinguishes the threshold for high and low tumor burden neurons. The beginning and end of each whisker stimulation event is indicated with a black dotted line. We plan to further interrogate this effect under the high burden stimulation condition by refining the temporal scope of our synchrony analysis. We plan to move towards a population level analysis, looking at correlations in smaller temporal windows, before, during, and after a single whisker stimulation. And we are interested in analyzing correlation patterns or changes in each respective population based on tumor burden status. In some concluding remarks, I want to say thank you to the amazing mentors and individuals who helped make this work possible. And a big thanks to those of you stopping by to listen. Thank you so much.